Okay, let's pick it up with where we left off. We're about to go into a discussion of in vivo protein folding. But before I do that, let's just remind ourselves that proteins fold because their sequences encode the proper information that allows them to collapse into structures that resemble the native state. And the rules that really govern that have everything to do at a gross scale with whether the proteins are dominated by long range or short range order. And remembering that in any individual protein, there's a unique set of interactions that hold the protein together and drive that process. But the rate of folding, at least, if nothing else, the rate of that folding reaction is determined by the organization in the protein. But let's watch a little movie now of proteins, of protein folding. And I think I give you this caveat during the movie itself, but reminding you that these molecular dynamics simulations of protein folding and other processes are themselves based on models. So they're only as good as the models that we have to generate them with, and beyond that, they're only as good as the predictions that they make that we can experimentally test. So I always give that caveat before showing molecular dynamics simulations. Here we go.
Okay, in this cute little movie of simulated protein folding, ab initio is just meaning from first principles. This particular protein doesn't really matter what it is, just a model system that a lot of people study. This particular study is using something called multi-scale modeling, which is just a coarse-grained look at the protein instead of considering all atoms. They simplify their model to a certain extent. They're assigning that secondary structure according to DSSP, which is just a secondary structure algorithm or assignment algorithm. And basically what's happening is that they're allowing the sequence to move around according to a set of very simple physical rules. And what we see is that something happens early on, which is a nucleation of a helix, and then we start to see some secondary structural elements, beta strands, flickering into and out of existence there, and we see this, the structure moves in and out of its organization, and then ultimately we see a nucleation of those secondary structural elements that will lead to the final organization of the molecule. And there they've noted what residues they think are of critical importance to that nucleation event. Looks like a formation of a hydrophobic core between that helix and those strands. And they say they've got more or less found the correct topology. They're using a term called molten globule. I might use that again in the folding part of this lecture. So that's a term you'll hear frequently for describing a partially folded structure. And then they've overlaid several partially folded structures from this simulation to see what we more or less wind up with. And then finally, in the end, they take an average of those, and it looks more or less like the crystal structure. So that to them is an indication that their model system, their experimental method has worked, and that they have defined the folding pathway of this particular protein. I will just add here that there are a variety of other groups doing protein folding simulations. This particular movie isn't from a group that I think is really reputable for some reason. It's just a cute movie. Most of these show more or less the same phenomena, and I think their utility is variable, and they serve as a cute illustration. It's not always clear how these models serve as predictors for real experimentation, but they certainly do show that with the simple set of rules that we have available, we can make proteins fold up in a computer simulation.
Now, back to protein folding. This is another version of that sort of funnel-shaped conformational energy landscape, and reminding you that basically in the x and y axis here we've collapsed the degrees of freedom available to the chain into 2 dimensions. So really, this should be a hyperdimensional space, but we're making it 3 dimensions. And then in the third dimension is just energy. And again, unfolded proteins, high degree of conformational heterogeneity, high energy, and then as we restrict that heterogeneity, we begin to populate folding intermediate structures, partially folded states perhaps, and the native state is in there somewhere. This particular diagram, taken from this great Nature of Structural Biology review that's fairly recent, breaks it into intramolecular folding, single domain proteins, single chain proteins, and then intermolecular protein folding involving oligomerization, aggregation into amorphous, unstructured precipitates, perhaps, and formation of amyloid. Some of these can be biologically useful, right? The oligomerization of a protein might be coupled in some way to other folding events. Amorphous aggregates, right? Fluff in the bottom of your test tube, cataracts in your eye, versus amyloid fibrils, which are highly organized structures, as we'll see. Both of these can lead to toxic effects and disease. In any case, this cartoon sort of captures many of the aspects of protein folding that I've already described, as well as some that we'll go into that are more relevant for protein folding in vivo. And again, I just want to make that distinction between protein folding in vitro in an experimentation when we have a homogeneous sample and the way proteins behave under those conditions versus the way they behave in the crowded and complex environment of a cell. So I'm going to talk about chaperones in this next section of the lecture, and I'm not going to talk about some isomerases that are also critical for protein folding, but focusing on chaperones for a moment, they help a protein adopt the correct fold, right? That is to say, de novo folding or co-translational folding, right? The protein's coming off the ribosome, chaperones are helping it fold, but they also play a role in the refolding of a protein or the prevention of aggregation of unfolded proteins after some stress event. I'll say it again, but here let me state that the chaperones that we're about to talk about were first described as heat shock proteins, so proteins that are expressed in response to cellular stress. Those stresses, of course, inducing unfolding of proteins, which can, again, in that crowded cellular environment, can rapidly lead to aggregation and precipitation, a bad endpoint. The isomerases, which I'm not going to talk about, we can think of two kinds of isomerization which would be obviously important for proper folding of proteins. One is isomerization of prolines. Sometimes, not frequently, but occasionally, peptide bonds form. You form cis peptide bonds, let me back up and say that again. Most of the time, proline exists in a trans conformation. I'll take you back to a couple of lectures to think about the cis-trans isomerization of prolines. Occasionally, prolines exist in a cis configuration, and in those cases, sometimes an isomerase is required to shepherd that proline into its proper folded state configuration. You could easily imagine that an improper configuration of a proline will dramatically alter the structure of that protein. So to achieve the folded state, sometimes we need some help. Proline isomerases play a role there. Likewise, disulfides. Disulfide isomerases can help reorganize the disulfide configuration in a protein so that we adopt the proper set of disulfide bonds. You could easily imagine a case as that ribonuclease example that I gave in the beginning, that if our sulfhydryls are interacting with the wrong partner, then we're not going to be able to achieve the final folded state. Now, what Anfinsen saw is in the case of ribonuclease, the molecule can be reduced and then reoxidized to its standard set of disulfides without any assistance, but that's not always the case, and certainly not always the case in vivo. In any case, on to chaperones. Not quite chaperones. Let's start with what happens when we make protein. So proteins are made on the ribosome. We've talked about the ribosome in the last lecture in terms of ribosomal RNA. We know that the ribosome is a machine that translates RNA information into protein. And you could imagine that as that chain emerges from the ribosome, the molecule has to begin to fold somehow. If it's true that the sequence information itself contains the fold of the protein, then we should expect to see the protein begin to fold as it's translated. And that does in fact happen. This is an NMR experiment. I've shown you two-dimensional nitrogen proton spectra several times, so they shouldn't be too confusing to you. This is a very recent review article that shows how proteins fold co-translationally. What do I mean by co-translationally? In the left-hand spectrum here, we're looking at the molecule. This particular molecule, it happens to be a myoglobin molecule. The first 105 residues are translated, and then translation is arrested or stopped. There's a particular sequence signal they give to arrest translation. And what do we see with these first 105 residues? Well, it mostly looks like that unfolded mess that we saw. And to draw your eye, they've drawn these circles around different parts of the spectrum where we expect to see the folded state, the signatures cropping up. And in fact, we do see some. If we look closely, we can see evidence for the folded state of the protein, even this early on. As we let the molecule proceed a little bit farther, so after that first 105 residues, we allow translation to go another 90 residues or so before we arrest translation again. Then we see yet more evidence of the folded state cropping up. And in fact, we would say that in this particular structure, we are more or less populating that folded structure on the ribosome. After release from the ribosome, then here's the native structure, and you can see why they highlighted these particular residues, right? You can see what those structures look like, and it's clear to see that we're beginning to form that. Maybe this is one of those molten globules that I was describing in a previous slide. And then after we move the chain along a little bit farther, we go a little bit farther along in our folding pathway, and then we fold up entirely. And here's a cartoon that explains that same process for each of those spectra. Important to remember in this case that there are no other, in this construct, there are not other molecules that will assist the protein folding. That's going to come back again, though, as we'll see. So keep that in mind. This is just the ribosome and the protein being translated. What these studies of co-translational folding tell us, though, is that the folding rate is dependent on codon usage. So that tells us that depending on the codon usage in our message, the molecule is going to be translated more or less quickly, and that's going to affect the ability of the protein to fold, the rate that the protein will be able to fold at. What else do we know? Well, we know that the exit tunnel off the ribosome is fairly rigid, and therefore that the chains, we call these the nascent chains, they're being born off the ribosome, must fold outside the ribosome. And the data that I just described to you shows that partial folding occurs even before the chain is completely synthesized and released from the ribosome. What the data don't show you is that chaperones, I mentioned that word chaperone a moment ago, chaperones assist the folding on the ribosome. Not in this particular case, but in other cases that we'll describe in just a moment, chaperones play an important role of making sure proteins that are translated are immediately allowed to fold to their proper structures. Sorting complexes shepherd nascent chains to their cellular targets, so there is a complex machinery that takes those nascent chains and delivers them where they need to go.
So because we're talking about protein folding and protein folding on the ribosome during translation, I wanted to revisit this video, and I'll just show you a short snapshot of it, pointing out a couple of the important ideas about folding on the ribosome that are clear from our current understanding, current research. So let's start that up again. Right, so here's folding, and I'm going to point out when we begin to, so there's the tRNAs moving across, there's our nascent polypeptide coming around, and you see that there's a tunnel that it's emerging from, and that the chain is very long and extended. We're probably not getting a good sense of the dynamics there, but what we show happening now is that the protein is trying to fold up into some compact conformation, and we see that happening. The biggest thing that's missing from this picture, of course, now we move back around again, and you see the polypeptide still growing and about to be kicked off. There it goes, floating away. The big thing that's missing from this picture, of course, is the presence of chaperones that are important for the shepherding of that nascent polypeptide into its folded state. So those aren't drawn on this cartoon. The other thing too that I want to make very clear is that I'm going to back it up again and show you this long extended chain that we're seeing is probably not terribly accurate. The chain is very dynamic and very flexible, not extended like this. So we're not seeing much in the way of chain movements here that are at all realistic. And of course, that folding probably isn't going to occur without the presence of some chaperones to help it along. So, but you still get the idea. A very nice little cartoon there. Right here, we see that tunnel that we know the nascent polypeptide emerges from before it begins to fold. Some of the experimental data out there suggests that while the tunnel might be fairly dynamic, it's not dynamic enough to allow protein folding inside the ribosome. So nothing's happening until we emerge, and then we start to see the chain emerge, and the data show us that the chain is very dynamic up to a certain point, and then it starts to become a little bit less dynamic, perhaps as it starts to adopt these structures, begins to adopt folded structures. Some of the data I show you as well suggests that we begin to adopt native-like structures fairly early on. So that's important to note as well. And again, that chaperones are important for many proteins to fold off the ribosome.